Hello and welcome to OMF's first virtual open house 2021. Unfortunately, due to the pandemic, we're not able to host an in-person event this year, but I'm so grateful with this Zoom technology. It gives us the platform to share a research update with you, and in fact, it allows more people to join us. So we'll be sending you a recording of this soon after the event for you to review later or share with others. We strive to provide timely updates from our collaborative research network as soon as they're available. And because so many of our foundation staff are patients or caregivers themselves, we hold a personal stake in our mission to end ME/CFS. And we only fund research that we think could lead to promising outcomes. We're honored to host this open house to update you on OMF's current funded research projects. And most of you know us, but for those that may not, to give you a little background of who we are as an organization. My name is Linda Tannenbaum, I'm CEO, and I started Open Medicine Foundation in 2012 after our own daughter was diagnosed with ME/CFS. And after seeing 20 or so doctors, I realized there were no diagnostic tests or treatments for this disease, which was preventing our daughter from leaving her bed and living her life. My goal in creating OMF was to change the landscape for those suffering with ME/CFS by doing whatever I possibly could to find a way to diagnose, treat, prevent, and ultimately find a cure for ME/CFS. It was obvious that funding and spearheading open, collaborative, global research was needed. Since we started it in 2012, we've grown immensely as an organization, and we now lead the largest international nonprofit research effort for ME/CFS and related chronic complex diseases such as post-treatment Lyme disease syndrome, fibromyalgia, and most recently, post-COVID syndrome transitioning to ME/CFS. OMF has raised over $30 million and facilitated the establishment of 5 prestigious ME/CFS collaborative research centers located at top universities at Stanford, at Harvard, Uppsala in Sweden, Montreal in Canada, and our newest center in Melbourne, Australia. For this event, I am thrilled to be joined by the directors of our 5 OMF-funded centers, Dr. Christopher Armstrong, Dr. Jonas Bergquist, Dr. Alain Moreau, Dr. Tompkins, Dr. Wenzhong Xiao, and Dr. Ron Davis. Each director will share details and updates on their existing OMF-funded research projects powered by your generosity and our community. Without further ado, please welcome our event moderator, Dr. Maureen Hansen. Dr. Hansen is a brilliant member of our 18-member scientific advisory board, a professor in the Department of Molecular Biology and Genetics at Cornell University, and director of the Cornell Center for Innovating Neuroimmune Disease. Dr. Hansen has conducted groundbreaking molecular analysis of many facets of ME/CFS. I'm so delighted to have her with us for this event. Thank you very much, Dr. Hansen.
Thank you, Linda. Hello, and welcome to OMF's virtual open house. I'm delighted to be here. My name is Maureen Hansen, and I'm proud to be one of the members of OMF's Scientific Advisory Board. Over the years, I've been able to watch OMF grow and progress as an organization. I think what makes this organization really unique is the focus on both innovation and collaboration. Innovative thinkers help us speed up progress, and collaboration brings leading scientists together to openly share ideas and work in a joint effort to end ME/CFS. OMF's ultimate goal is to improve patient outcomes. OMF wants to generate research that provides the ability to deliver patients better diagnoses, treatments, and personalized care. OMF aims to do this through an open research model to understand the molecular fundamentals of disease. Today, you will hear more about this collaborative research model from the directors of the OMF-funded collaborative research centers themselves. Without further ado is Dr. Ronald Davis, chair of OMF's Scientific Advisory Board and the director of the ME/CFS Collaborative Research Center at Stanford. Dr. Davis is a professor of biochemistry and genetics at Stanford School of Medicine and is the director of the Stanford Genome Technology Center. Dr. Davis's history of interdisciplinary work, technology development, and unique approaches to previously unsolvable biological problems makes him an exceptional scientist to lead a collaborative effort to solve the mystery of ME/CFS. In addition, he brings a strong passion to this cause in the hope of finding a cure for his son, who suffers from a severe case of ME/CFS. Please enjoy this update from Dr. Davis about the latest research into ME/CFS happening at Stanford.
Hi, Ron. Thank you very much for coming to give an update for Open Medicine Foundation today.
Glad to do it.
Obviously, it's been a very interesting year this past year, and a lot of things that we've been working on have been kind of progressing in their own way. I think, obviously, COVID-19 has had some interruptions, but also created some opportunities. I know from some of the work that you're doing, it's quite interesting in its relationship to infection. I'm speaking specifically of looking for the genetics within pathogens, or for genetics from pathogens within patients. So I thought maybe we would start with that, and maybe you could just give a little description of what that project's about.
Well, the patients look like they have an infection. The way they behave, and the fatigue happens with an infection. So it's always been a question. Well, maybe they do have an infection, but you'd like to have evidence for that. And so one way to get that evidence is that these organisms will be killed by the immune system at some level, usually. And when they get killed, they release their DNA or RNA, depending on what the organism is. And then you can find that in the bloodstream. Now, of course, with the patients, we can only take out so much blood. And so you have to worry about in 10 mils of blood, how many DNA molecules or RNA molecules are in that. And so you have a certain sensitivity. We would like to develop some technology where you take blood out, pull out the DNA, and put the blood back. That way you could go through and recover a large amount of material and make the assay more sensitive. So when we come up with a negative result, it doesn't mean there's not anything there, but it means we can't detect it. So the first pass was the herpes viruses would be a major candidate. So we've developed a technology that allows us to simultaneously amplify up different regions of the organism. And we do about 10 different regions of each organism, and we do them all simultaneously in a single tube. That's called a multiplex reaction. And I have Badong Shen is a real expert. He's trained as a chemist. He's really good at this kind of stuff, and worked out a whole new simple technology that's very inexpensive to do this. And so we can look at he's set up to look at 20 different DNA viruses that are the sort of primary candidates that we think might be present and causing a problem. But recently, he's also done 17 RNA viruses. Enterovirus is one that's been implicated in ME/CFS in the past. So that's just, that work has just started. We've done about 100 patients and 100 healthy controls with just the DNA viruses at the first pass, and we'll start doing the RNA viruses real soon. But so far, what that is indicating to us is that we don't see any obvious major amplification of these herpes viruses and other DNA viruses compared to healthy controls. We really need to do more patients. Also, patients sometimes say that they think they have a reactivated virus. And so we'd like to get a connection with the local patients so that we could actually get a sample from them when they think they have a viral infection, and see if we can confirm that. Again, that confirmation is to the sensitivity that we have. But we can certainly see it in healthy people. These reactivations occur. In most cases, they have virtually no symptoms.
Yeah, I mean, that's fascinating. I think I know that this technology is also going to be utilized within this COVID-2 ME project that a number of the collaborative research centers are working on. I know that you're going to be interested in determine if there are any viruses as opposed. I mean, obviously you'll have the COVID virus, but as in addition to that virus, other pathogens that may be reactivated during that infection phase when they're collecting those samples from within the hospital. So I think that will be obviously quite interesting. Do you have any thoughts to add?
We've done a small number of those patients, I think like 6, and it's a matter of getting access to the samples. And because what could it be? It looks like the COVID patients, some fraction of them are not recovering, and they're called long COVID. And it looks like from their symptoms and also some of the physicians, they're diagnosed, clearly diagnosed with ME/CFS. So our guess would be maybe 10% of the COVID patients will be coming down with ME/CFS. So what is possible is that the COVID doesn't create it directly, is that it activates something like EBV, which we know can cause it. And that activated EBV is what's causing it. That's important to know, because if that's the case, then you could be treating these patients with anti-herpes virus and drugs to maybe prevent that from happening. But we need to do it quickly, because there's millions of people who've already had the COVID. Anyway, it's been really tough to get the government to come up with grant money to do that. Anyway, the other thing is that this could also be a parasite. They're hard to analyze and they're hard to spot. So, and doctors will say, "Oh no, you don't have a parasite." Well, they don't look.
Yeah.
Right. So, and why don't you look? Well, because you wouldn't have one. Anyway, so we've made probes for lots of the common parasites that are particularly bad. That also could last a long time. And one reason for doing that was that the symptoms of sleeping sickness are very similar to ME/CFS, and so that's caused by a trypanosome. So we put in probes for all the different trypanosomes. That's just guessing, right? That might be possible. And then we've also done probes for the ribosomal region. We'll sequence up the ribosomal region and for all the bacteria and all the funguses. Again, that's just giving us an idea of what might be there, and that's first pass.
So do you?
So that's ongoing, and we'll, as soon as we were ready to do this when the pandemic happened, and then with the pandemic, you can't have patients come in. All the clinics were shut down. The ME/CFS clinic at Stanford was shut down, and they only allowed COVID patients to be there. So unless it was an emergency. So we're trying now to reactivate and bring patients back in, and also get connected with some of the other clinics that have patients to get samples.
I mean, obviously there has been some obstacles from this pandemic. We all know that. In terms of some of the opportunities, I guess, we are doing this collaborative study. We are getting samples from people who have COVID during the significant phase of their infection, which is actually quite difficult to get, hold of samples from people at the initial stages of a significant infection. So that, I guess, those samples are presenting an opportunity for you to identify, I guess, whether there are going to be these triggers. Yeah,
that's correct.
And so in terms of this type of technology, how do you think it relates to, I guess, the general pathogenesis? Are you thinking of this being interesting in terms of a trigger, or something that could be continually maintaining ME/CFS as a disease?
Well, it's possible why the disease lasts so long is you get reactivation of things like ABV or HHV6, and that kind of starts the whole disease all over again. And so that could keep people in it. And that's one of the, why do people not get well? You usually get a disease or an infection and you get over it, and you're not getting over it. And so a reactivation of a virus could actually keep you sick. That's why it's important to look at. And we'll continue to do that. It's very simple. It's now very cheap, and it's very easy to do. So, because the technology was set up to make it easy and cheap.
Yeah, that's definitely the hallmark of your group, getting fantastic technology cheap. I think that's testament to kind of the research that you've been doing there for a long period of time. Just interesting, I guess, talking about the pathogenesis of ME/CFS. Obviously, that's a good segue to talk about maybe the metabolic trap update as a potential other hypothesis for the disease. Where are we at with this metabolic trap? And yeah,
so the metabolic trap is, and there may be other traps as well. And so the thoughts are that something has happened to the patients where they can't get well. Otherwise, they would. So we're looking for something that can get into a state where you can't get out of it. And it looks very much like some kind of metabolic disease, and because you see such big alterations in metabolism. So that's why we've been looking at metabolism a lot. And so one of the possibilities is what happens right after when you have an infection, and that's what we have to look at because that's what starts the disease, is that tryptophan is released from the albumin, and that tryptophan then floods and is taken up by a class of immune cells. And then those immune cells convert the tryptophan to kynurenine, and that process of making kynurenine inside of these immune cells allows those cells to regulate the immune system. And that pathway can allow them to suppress things like autoimmunity. And then that's what cancer does. It activates this enzyme and makes kynurenine, and that makes the cancer cells resistant to attack by the immune system. So the kynurenine production seems to be key for the immune system. Now, a mistake has been made by a lot of the investigators in their thinking, and that they say, well, that can't possibly be the case because the liver makes kynurenine. Yeah, that's true, but that kynurenine cannot get into the immune cells in sufficient amounts. Now, that's something that Rob Fair, who came up with this idea, he can model all this, and he can model this in terms of modeling it in terms of the transport of kynurenine into cells. It has to compete with the amino acids, and it doesn't compete well. So you really cannot get kynurenine from the plasma into these cells. It has to be made in the cells. You have to import tryptophan, which does get in effectively, and then it can get converted to kynurenine. So,
so it sounds like obviously, the technology, or to test this hypothesis, is going to involve utilizing cells and looking at metabolism within the cells themselves.
Right. So what's unusual is the fact that this enzyme that's made from the IDO1 gene, which does the conversion to tryptophan to kynurenine, is unusual. And this is a paper published in 1967 that Rob Fair knows about. And that shows that this enzyme is substrate inhibitable. Very unusual. So in other words, the substrate that it normally uses, if that substrate gets too high in concentration, it inhibits the enzyme. So if you were to inhibit the enzyme, that means you can't make kynurenine. If you can't make kynurenine, you can't regulate. And the only way to go back to being able to regulate is you got to get rid of the tryptophan. Well, the only way to get rid of the tryptophan, as far as we can model it, is for the enzyme to be active. But it's not, it's substrate inhibited. So it could spontaneously, in some sense, sometimes reactivate enough to get the tryptophan down, and then you would presumably get cured. But the probability of that is extremely low. And in fact, you do see some patients that do get ovaries, but not many.
Yeah.
So that's why we call it a metabolic trap, in the sense that it can't get out of it because it needs the enzyme to get out of it. So the question is, that's just you can see this in the test tube with purified enzymes. The question is, does it actually happen in the cell? So we've done two different things. One is that we've engineered yeast, which is very quickly to be engineered and work with, and we have a lot of expertise in yeast. So we put the human IDO1 gene into yeast, and we put it under control so we can control the level of it. And then to see whether or not it works, the kynurenine that it will make from tryptophan can be used to make NAD, which is a very essential component for survival and growth of the cell. There's a number of ways to make NAD, so we've deleted those from the genome of this yeast. So the only way it can make NAD is to make kynurenine. And to make kynurenine, it has to turn the gene on and convert tryptophan to kynurenine. So if we put that yeast into high tryptophan concentration just in growth, it won't grow. It stops growing.
Wow.
And if you put in kynurenine, it doesn't get taken up super well, but the yeast will then start growing. So it shows that the block is at the kynurenine level.
That's excellent. So you've proven, like, I guess you've shown that this trap does exist. So from the theoretical to the real. But now, how do you plan to kind of transfer that to determine whether this trap is existing within ME/CFS patients?
One other interesting little connection to that is that if we then remove the tryptophan and let the yeast cells sit there for a while, eventually they start growing.
Oh.
And then we look into yeast and we realize that there's a couple of other genes that will convert tryptophan to other amino acids. In other words, they consume the tryptophan, which is what you have to do to undo the block. So we've simply deleted those genes. Now you do it, and now the yeast don't grow.
Okay.
Right. So that's also kind of confirming the idea that if you get rid of the tryptophan, you can start growing. Now, the value of that system is two things. One, it shows that you can do this in vivo in the cell. You can initiate the trap, and it does exactly what we expected, but like it is in the test tube. But with yeast, what we want to do, and that's why I talked about the fact that you've got to get rid of the tryptophan in order to grow. That's also what you want to do if this trap is what's causing the disease. We have to reactivate the IDO1 gene product, the protein, in order for it to convert the tryptophan to kynurenine and get rid of the tryptophan. So that might be done by some existing drug. It would bind to the enzyme and prevent it from being inhibited. In other words, it would inhibit the inhibitor. Now, that's just a screen. Now, you can do those screens in test tubes and all that, but yeast makes it really a really simple thing. You simply take a large plate, put in lots of tryptophan, get the cells there, and they're now blocked, but they're sitting on the plate. They're not dead. They just don't grow. And you simply spot a little bit of compound on those plates. And you can actually, you could even set up an automation system to spot those come up. We have all of the FDA-approved drugs in our freezer because we actually work with all of them and looking at the effects it has on yeast. So what we want to do is look at different concentrations. So we're not interested in the concentration that the drug is used in humans. In fact, you have to use a much higher concentration in yeast just to get it in, because yeast doesn't is a barrier to most compounds. And so you simply put a solid amount of the drug at a spot. It then diffuses, and as it diffuses, it lowers its concentration. And what you look for is a ring of growth. So it is at a concentration now that will inhibit the inhibition, so to speak. And you can see that even though right near the spot, it may be too high a concentration, and that drug itself inhibits the growth. So you don't have to do a whole bunch of experiments at different concentrations to see which one works. You just look at the plate and see if you see a ring. Your eye is really good at seeing that kind of thing. So you could do a large number on each plate. And
Wow,
so
that's fascinating.
you could go through the entire collection of FDA-approved drugs fairly quickly and cheaply. That's also a very cheap thing to do. We're just trying to set that up. We also have robots that we can do these things and do growth curves and it's that sort of thing. Probably I would prefer to do a few of those as well as the plates because we don't want to miss something. But also we can do a much finer analysis of what concentration to use. Then also we've done this with Rob Fair is in charge of doing that. He's working with some of our really good technicians. We have some fantastic technicians, the best I've ever seen. And they're technicians, they're not PhDs, but they're operating at the PhD level. And they have set up taking immune cells and converting them to monocytes. And then those cells have the pathway on IDO, have the IDO1 gene active in those. Now we have to stimulate them to produce the enzyme, and LPS is what's often used because LPS activates this whole thing, and that's what mimics an infection. And then if you then put in lots of tryptophan, and these experiments are done by first inhibiting the enzyme, so it allows the tryptophan to accumulate. And then you wash out the drug, but keeping them in high tryptophan. They are blocked. And so it looks like the metabolic trap works in human cells as well. Now, those will be a good source for other kinds of experiments looking at it. And also, can we wash out the media from the tryptophan and still keep them trapped? They shouldn't export the tryptophan very effectively, but we don't know for sure. And so those are the experiments that are now going on. Now, those experiments have happened recently. Those are just cells coming from the blood bank because we were not allowed to have patients into the lab to take a blood sample. And so we're just now getting back to being able to have patients in the lab. And now we desperately need to get patients to come into the lab now. We've been doing a lot of experiments with archived frozen cells that we've had, that when we do a blood sample, we prepare cells, we do a few experiments with them, and anything left over gets frozen. So we've had a large archive of ME/CFS cells, and that's been running the immunology with Mark Davis for a long time now. But we're running out of frozen cells. We've got to get new, fresh samples in. So I'm encouraged by this. We don't know if the metabolic trap is what's causing the problem, but it is certainly feasible, and it fits the sort of the biology. This would happen after an infection, and that's what we have to look at. What's causing ME/CFS? Is it often an infection? And it's involving immune cells. It's involving immune cell regulation. Kynurenine is a major regulator. Kynurenine in the brain suppresses inflammation. So there's just a lot of correlations with this behavior of this pathway and what you see in the patients. So it's sufficiently connected that we really need to explore it.
Yeah, I mean, it sounds like a definitely very, very interesting area, and a lot of people are very interested in it. So thank you very much for giving that really lengthy update on that.
Speaking next is Dr. Ronald Tompkins. Dr. Tompkins serves as the Chief Medical Officer of Open Medicine Foundation, as well as the co-director of the OMF-funded NE/CFS collaboration at the Harvard-affiliated hospitals. Dr. Tompkins is a Sumner M. Redstone Professor of Surgery at Harvard Medical School, founding director of the Center for Surgery, Science, and Bioengineering at Mass General Hospital, and Chief of Staff Emeritus at Shriners Hospitals for Children in Boston. Dr. Tompkins' work in genomics and proteomics and their roles in inflammation and metabolism aims to discover the underlying mechanisms of NE/CFS. Please join me in welcoming Dr. Tompkins.
Thank you, Ron Tompkins, for coming to have an interview here today. I recall actually the first time that I met you in 2017. I think it was at the Stanford symposium at Ron's house after the symposium, or the community day, I think. And you weren't working on MECFS much at the time. I mean, I knew you were someone who'd done a lot of research in a lot of different fields, specifically around trauma. But I really remember that you had a really great understanding of MECFS from my perspective and the perspective of people I had collaborated with. I'd been working on MECFS for 7 years. Collaborators have been working on it for decades. And yeah, I was really impressed by that effectively. And so I guess digging into more of the work that you've done and more of the history of the work that you've done, and kind of get a better understanding of how you kind of came to that kind of understanding in regards to MECFS. I'm speaking, I guess, a lot about the glue grant, which I find a really fascinating project as one of the major things that you've accomplished in your career. And now we're in a pandemic 4 years later, and it's becoming all too more relevant again. So I thought I might start with that, chat a little bit about your work in the glue grant and how it may be relevant to this COVID-2ME project that we're doing collaboratively here.
Well, thank you, Chris. I recall our first meeting, and I was very impressed with a young man that was very interested in metabolism and a deep understanding about this particular problem. And I'm delighted that you've chosen to spend quite a bit of your career here trying to understand this problem. With Ron Davis, over the last few decades, we have explored a great deal in the whole field of injury research. And the glue grant was a $100 million project funded by the NIH to better understand from what's called genomic or gene expression perspective, as well as from protein changes, as well as metabolic features that occur when one is injured seriously enough to die. And the patients we studied had a 20% mortality rate. So it's very important to understand how the human system responds when you have a 1 chance in 5 of dying. It's about as severe stress as is possible. But in doing so, we had over 100 principal investigators and over 10 years to begin to understand and collect information in a very systematic way to how the human system responds in circumstances like that. I was very delighted that just getting together, we improved the survival rate by 50%.
Wow.
In major blunt trauma. And over the last few decades, our interest in the field of burns, we've almost eliminated the mortality in pediatric and children who are severely burned. The expectation is to survive. So I am very excited about the fact that if one takes science and really begins to apply it to clinical medicine, you can make a huge difference. My relationship with Ron Davis, who's gradually over time had to be interested in MECFS because of his son, has continually asked me about things as a doctor, for part of me as a scientist and part as a doctor. And I would just simply answer his questions. And both of us came to the realization that much of what we were learning in the area of injury was extremely applicable to this disease, MECFS. And so I've become more engaged to use that information that we gained after a tremendous experience in better understanding ME and how it occurs, and the inability to understand its etiology, I hate to use that word, but reason that it exists, and why doesn't it resolve normally?
I think that's the, and I guess I'd summarize that as looking at kind of that transition from the acute to a chronic disease state, and how that triggering component kind of interacts. And that obviously is super relevant to this COVID-19 research that we're looking into. But also it's probably interesting enough to talk about the common pathways, I guess, the body kind of goes through, or the stress pathways that it goes through in response to all varieties of different types of stresses. Obviously, you're talking about trauma or blunt trauma or burns in this case for the glue grant, but then you have infection and all these other ones. So what is, I mean, do we know that there is a common kind of path that is taken?
Well, let's just think about it, Chris. It's only been the last 4 or 5 decades, if you were incredibly ill, sick, that you would even survive. And that has purely been the result of our understanding of how to deal with critical care medicine. In the past, over our entire history, if you were incredibly sick, let's say you fell off a cliff and broke bones, you crawl back into the cave, you know, your family brought you maybe some twigs, leaves, and water, but you either survived or didn't. And it really was rather local injuries that actually allowed you to survive. If there were any terrible infection or terrible injury, you were left behind and eaten. And there was no, I'm just speaking from an evolutionary perspective, there was no advantage that genetics could provide that will allow you to survive. It's only been in the last 50 years that if you got that sick, you were going to be able to survive. So to come up with an evolutionary argument why I would respond differently to a terrible infection as opposed to a terrible injury is hard to make, because you would have simply died, and that you would not have lived long enough to reproduce. There is no evolutionary pressure that would come up and say, as a human, you know, I have a special response to infection. Yeah. It's just you would have simply have died. So it is reasonable that any serious stress and the recovery from it is rather common, more than different. I don't understand how you would have distinctions.
So that, I guess that's very relevant for the rationale we took for the looking into the COVID-19 to ME and using this kind of opportunity, I would say. And I guess it is a massive opportunity, not just because of the global interest, but also that you have the ability to identify people going through an infection right at the beginning of an infection, where usually there isn't as much attention paid to that occurring, and these people are popping up in the ICUs at hospitals. So I guess we can talk about that project and maybe give an update on how that's going.
This is the SARS coronavirus 2 is a very, what we would call pro-inflammatory virus, such that its infection creates a tremendous inflammatory reaction. And it can, in some individuals is pretty minor, but at least in the acute disease, and by acute disease I mean over the first 24 hours. What we're observing, however, is those who do survive, those who have been hospitalized, that if they're lucky enough to be discharged, that over the next 4 to 6 months, they're having persistent symptoms, very often pulmonary, but also other symptoms. And we're also seeing a second group of individuals whose original illness wasn't terrible enough for them to be hospitalized, but yet after 4 to 6 weeks, their symptoms not only don't resolve, but evolve and might even be worsening. So we're seeing this entire very significant population who, over the next 6 months after the original illness, have persistent and evolving symptoms. And the NIH has described that as post-acute coronavirus sequelae, PACS, PACS. It's really the same as long COVID, in which you just fail to recover from this horrendous stress. And unfortunately, it's not just restricted to those who had a severe illness. There are some whose illness was not that severe, and yet they are continuing to suffer, and if not even persist and get worse. And so I do think this is an opportunity to better understand what loosely has been termed post-infectious fatiguing illnesses, of which, in my opinion, ME/CFS is one of those. So because this is affecting millions, and to the point where at least the United States government is so concerned about it, they invested $1.15 billion in order to understand better, so that in the future we're not going to be surprised. So we have this 1.15 billion to better understand how this disease process evolves. And in my opinion, some of the symptoms of individuals over 6 to 12 months will simply be persistent damage to organs like their lungs or in the central nervous system or cardiac or other areas. And you can do normal blood tests or images and identify persistent organ damage. But at some point, those studies are going to become normal or not abnormal, and yet the symptoms are still present, which is very much what patients with ME/CFS face every day. They have very significant symptoms, yet the standard tests don't show abnormalities. And I think this is going to be a fabulous opportunity to be able to watch this process happen. And at 12 to 24 months, where you have blood tests and x-rays and all sorts of images, and there's no organ damage to blame, yet the patients are still suffering from this, I hate to even use the word fatigue, but it is an energy problem that if they exceed their threshold, they will pay a huge price. And it's difficulty focusing, their dysautonomic symptoms. And I think it is, I hate that it had to occur because of a pandemic, but I think it's going to be such a big issue that we're going to learn something from this.
Yeah, I would certainly say so. And I guess the study design we've gone for, and the reason why we're so interested in the ICU patients or hospitalized cases, if we can get to them, or even the non, is to get to them at the point when they've had this infection. And I believe that was to do with some of the work that you've done on that glue grant in the past, looking at trauma and identifying that some of the clinical trajectories, the different clinical trajectories from trauma over time, and the length of duration of recovery. Some of those markers were kind of established very early on in the infection phase or the acute phase. Is that correct? And is that kind of where we're at as well with the study? We're looking at the first week or the first week of infection effectively to look for these markers, and then following them on. to see what they may link to in terms of clinical recovery?
The aspect of the glue, there are multiple aspects of the glue grant that are very relevant. You can appreciate if you were hit by a bus that you end up in the intensive care unit. You also ended up probably with a lot of operations, but we do know we did learn a tremendous amount about how the white blood cells respond over time. And I can tell you that 80% of all the human genes respond in a maximum response over actually within the first day or so after the injury. So we've called that a genomic storm, because every single gene that could have responded, all human genes, 80% of them did respond in your circulating blood. Similarly, in each of our tissues, and the other tissues that we studied are ones that we could biopsy, and that was muscle and skin and fat. Because we were doing operations, we could look at individual tissues. But we used that as a reflection of what probably happens in all of our organ systems. And each of those had a very similar dramatic response. So we know a lot about how genes respond. And as a result of the genes, we also understand in separate studies about proteins. And we have for many years known quite a bit about how small molecules, how the molecular metabolic response occurs. And there is a very consistent coordination among all of those systems that occurs after injury. And it is our hypothesis or a thought that these are going to be very similar after very serious viral infections. And to a major degree, what we've observed in patients who've had a very serious COVID infection, it's a very inflammatory response. They're describing it as a cytokine storm. These are all things that we routinely see after injury. And it is our expectation that the genetics or genomic response is going to be comparable. Now, what happens then is you have patients who recover normally or rapidly, relatively rapidly. You can imagine being hit by a bus and you're not going to just jump out of bed, but it may take you a few months, but you will ultimately be normal. Be healthy, but it may take a few months, but that would be the very best human recovery possible. You can also imagine that there are individuals in which it's going to take them more than 6 months, but they will eventually be normal. And you would call that something like a delayed recovery, but a recovery nonetheless. And there are going to be individuals who not only are delayed, but never actually return to normal. So there is what we call an uncomplicated recovery. You were hit by a bus, but you did recover. And then there's complicated but recovery. And then there's complicated and you never actually make it all the way back. So those are basically 3 different types of recoveries, and you can associate those with genetics and with proteomics and with metabolomics. And it really might be useful in 2 ways. One, if you could use them for prediction, and you could very early identify those who are going to have problems so that you might treat them differently than others. And number two, you can take advantages of those differences, and it's possible they can become targets for treatments, so that it's potentially those targets could be used as therapeutic targets to improve. So those are the major motivations to take what we know with injury and maybe apply some of those concepts in this possibility of development of ME after this terrible infection.
Yeah, I mean, that's really fascinating, and I think a great rationale for the reasons for doing this study. I kind of want to go over a few of these other projects that you've been up
Sure.
doing. I mean, I think one of the more fascinating ones for me, and probably one of urgent need, is the PASS-CAT program. Maybe you could do a little brief introduction of that, and we can talk a little bit more about that.
The PASS-CAT is, and let me try to remember what PASS-CAT stands for. It's a patient assessment of symptom system, so that it's a computerized ability for patients to take their time that would very carefully characterize whether they have what I would call a sentinel symptom that the doctor should be aware of. And so let's talk about a couple of symptoms of ME that are very characteristic of ME, that if present, the doctor should stop, listen, and pay attention to. And I think one of those is post-exertional malaise. And I mentioned it a little bit, but with expenditure of energy, patients know they're going to pay a price for it. Number one, they have a threshold, and if they remain below that threshold, they can survive reasonably. But if they cross that threshold, they're going to pay a tremendous price for it. I know this is loosely termed as fatigue, but this is a very more specific form of fatigue, and it's not something that doctors are traditionally aware of or pay attention to at all. I call it a sentinel symptom. And if that type of symptomatology exists, the doctor should stop, look, and listen and do something about it. I don't know what the problem is, but it is a symptom that should be, it's not in your head, it's not psychosomatic, you're not depressed, it's real. And so the other part of it is CAT, and CAT is computerized adaptive testing. And what it has to do is, it's been used in testing methodologies all over. If you do certification exams and all sorts of computer testing, it is an ability to take a database of questions, ask you a few questions, and if you're competent in areas, move on to other questions. And in a very precise and prompt way, identify your level of competence in many different domains for certification. And it's, for example, in this context, if you don't have headaches, don't ask me a page of questions about headaches. And but if I'm having pain, let's get to the point of what kind of pain I have. And computers can decide and to understand and come up with questions much like your doctor would. If he or she were sitting across from you, they won't ask you a million questions about headache if you don't have headaches. Yep. But they will take your symptoms until they're comfortable that they know what kind of pain you might have. And there's a computerized way of doing exactly that. And it will allow patients to come up with their set of symptoms and a report for their clinician. And the way we're looking at it is we would like a sensitivity of 95%, which means if you have one of these symptoms, we're going to do our best to not miss you. there's less than 5% chance of missing you, but it'll be about an 80%, if indeed you have one of these symptoms, there would be about an 80% likelihood that indeed you have the symptom that we're looking for, like post-exertional malaise. And it would tell the clinician, you have a very important symptom. It needs to be evaluated. It might be simple, and we have suggestions about evaluation and treatments, or it might be that 20% where you really need to see a specialist. But this is really aimed at the primary care physician. Number one, it tells them this is not psychosomatic, you're not depressed, there's something real here. These are things you need to do to evaluate it. And these are the indications for referral to a specialist and what kind of specialist. And these are some suggested treatments. And in the long run, we can use them as quantitative metrics for intervention studies.
Yeah, I mean, I think I really like your description in terms of like asking you a lot of questions regarding headaches when you don't have headaches. But I think it's interesting because rather than having 100 questions in surveys about all the different types of symptoms they may have in all different ways, effectively what you're trying to do is get to one symptom and then deeply characterize that one symptom. Even the idea of fatigue just being one way of saying, oh, I have fatigue, rather than kind of getting to a better understanding of the specific aspects of it, which is like the qualitative. I mean, everyone has their own experience, but it's probably unique, but we might term it all a very similar thing. So I think that's fascinating that, this program and the way that it operates, using the computerized adaptive testing is a very clever way of trying to get deeper information than we're currently getting on patient symptoms.
MECFS is such, if you look at it, it has such a dynamic range. At the one end, you could be bedbound. A less severe, you can be homebound, but you can't work, or you can be homebound and be able to work, or you can barely be functional at work, but you have no other life. Yeah. I mean, it has a dynamic range that is unbelievable.
So this is where we need to get to. And the diagnosis components for research, not just for the patients, but for research, has been a complication, I guess, because of that dynamic range, but also the variation in which people get diagnosed and all sorts of things. So this would really help, I think, in many different capacities. So I'm looking forward to that program, or this PassCat being ready for use. I also want to kind of touch over some of the work that you're doing in the imaging sphere, the neurological or neuroinflammation sphere. Did you want to kind of go over some of that work just briefly now?
My interest in the neuroinflammation, it has been long-standing with colleagues in neurosciences and in nuclear medicine here over the last 3 or 4 decades. Part of it was related to understanding dementias, but what has become abundantly clear over the last 10 years is that the role of inflammation, neuroinflammation, role of glial cells, impact on astrocytes, and it's what occurs within neuronal reduction in synaptic connections, apoptosis. I know these are all very technical terms, but it is so critical to the understanding of how the brain works. And we've gathered so many wonderful tools along those lines, and there's so many people here at our hospital very much involved in that. So, and as I look at MECFS, this issue of difficulties with cognition, with sleep, circadian rhythm, so-called brain fog, which is such a complicated matter. it just, to me, the role of neuroinflammation is so important. And until told otherwise, my presumption is it exists and is part of the etiology here. And we have wonderful tools for this, both in the area of MR and MR imaging, as well as positron emission tomography, developing these molecules to image and identify these findings. So it just makes sense for us to pay attention to this and use many of the things that we have already fully developed for dementias, for Parkinson's, for ALS, for so many other neurological diseases. And we have wonderful colleagues here who have been following patients with ME for decades. And it just makes sense for us to explore these possibilities with so many of our imaging individuals, imaging colleagues, and our neuroscientists. We have a huge neuro group that we've gotten to be very interested in the topic.
Yeah, I mean, obviously with all that expertise, you can see that there's a fantastic kind of, I think there's probably a fantastic resource there, but also it may lead to kind of interesting treatments and repurposing of treatments, I guess, for ME/CFS, if you can find some commonalities there between different neuroinflammation paths.
There's many companies here that are very interested in very expensive monoclonals against so many targets, but there are also a number of companies here in ALS, and for that matter, in other neuroinflammatory diseases like dementias, repurposing drugs that are anti-inflammatory.
Yeah, that's fantastic. I mean, that's these
safe that should have very relevant effects, reduction of neural inflammation, but they're safe and not overly expensive. But of course, we are going to have those with those. We have many companies here that are interested in the expensive drugs, but it also, there are many technologies and opportunities developed because those companies are interested in the expensive drugs. So I think there's a good interplay and opportunity.
Yeah, I mean, that would be fascinating. I mean, I think we've covered a lot of major topics in the world of ME/CFS. We're talking etiology, your projects, and diagnosis, and maybe even looking towards treatment avenues towards the end. I think obviously you're doing a lot of fantastic work there at MGH and Harvard Associated Hospitals. I'd like to thank you very much for coming and giving an update. Yeah, I think people will be very interested to kind of hear more at some point in the future.
Sounds great.
Thanks, Ron.
Next up, joining us all the way from Sweden, is Dr. Jonas Bergquist. Dr. Bergquist is the director of the MECFS collaboration at Uppsala University in Sweden. He's also a full professor in analytical chemistry and neurochemistry in the Department of Chemistry at Uppsala University, adjunct professor in pathology at the University of Utah School of Medicine, and distinguished professor in precision medicine at Wenzhong Medical University in Yantai, China. Dr. Bergquist studies numerous conditions, including neurodegenerative disorders. His research into MECFS is focused on characterizing the neuroimmunological aspects of the disease using proteomics and metabolomics, with a special interest in cerebrospinal fluid studies. Please enjoy his brief presentation on research underway at Uppsala.
Hi, Jonas. Thank you very much for taking the time to do an interview update for Open Medicine Foundation and the audience here today.
Hi, Chris.
Hello. First, obviously it's been a very crazy year this past year, but I thought maybe we should start with a very recent paper that you had published in regards to a hypothesis relating to ICU and recovery and how that looks to be a similar mechanism, or you hypothesize to be a similar mechanism to MECFS.
Yeah, that's a paper we published in Frontiers in Medicine recently, and we are there trying to allude to that the symptoms and the situation we see in intensive care unit patients that have been critically ill and been through quite severe medical treatment also, in some cases end up with symptoms that are quite similar to what we see in MECFS patients. And now we are trying to learn from these mechanisms that we have been studying quite well in intensive care unit patients. So to make the story a bit possible to grasp, I think we should maybe just focus on a few things that we are looking at. There's plenty of information in that paper. So as you all know, probably we have a lot of different endocrine glands in the body, endocrine organs that signals to the rest of the organs in the body and also controls a lot of our biological functions in the body. And what we know is that some of these adrenal glands and pituitary glands and hypothalamus, etc., parts of the central nervous system will be affected during critical illness. And they will stop sending out these signals that we need to actually regulate our normal functions in the body. Or they will not have the rhythm that these endocrine organs need to have to actually control our normal functions. So our idea is now to try to see if we see the same pattern actually in our MECFS patients. So initially what we will focus on is hormones like the steroid hormones, sex hormones, thyroid hormones, and some of the neurotransmitters that are also released by these adrenal glands and pituitary gland and hypothalamus. So I think we can actually learn something from this and use the similarities, but also the differences we see between critical ill, intensive care unit critical ill patients and the MECFS patients.
Yeah, that's fascinating, and this is the type of research that we really want to kind of see transition to MECFS, taking it from other diseases or other types of illnesses, other mechanisms, and seeing how that could relate to this disease. And obviously it's very relevant at this time as we speak of the COVID pandemic and all sorts of different types of illnesses that are coming from that, the long COVID, and how that relates to MECFS. Obviously, Open Medicine Foundation has recently funded research into looking into COVID-2ME. And I know a large majority of that work is happening there in Sweden, or at least a large majority of the collection of samples. I thought maybe it'd be great if you could kind of bring the audience up to speed on that.
Yes. So what we are doing now as a cause of the pandemic, actually, we have a unique opportunity to actually collect and follow patients where we have a known onset of an infectious disease, a viral onset, And we are now able to monitor over time what happens in those patients. And as we already now see, now it's 1 year since the pandemic hit Sweden. I think we have now a chance to actually see how patients are going to react and what kind of symptoms that will be long lasting after the infectious disease in some of the patients. Not in all, of course, but in a quite large percentage of the patients. So what we try to learn now is, when we know we have an ongoing a clear onset of a disease, like an infectious disease, what happens in the body? How does the body cope with the situation? And when it comes to COVID, we know that some patients will have neuroinflammation. So actually, even viruses will transport into the central nervous system and also get quite severe neuro-related onsets. And some of these patients end up in our neurointensive care unit for treatment. While others have so much milder symptoms, they don't get, they feel they are sick, but they have like a mild flu maybe. And still, when they have recovered from that initial infection, they still have ongoing sequelae, as we say, symptoms afterwards. And those are, to a large extent, very similar to what we see in our ME/CFS patients. So this is what we're trying to use now as a, sort of unfortunate situation, but also a possibility for us to actually see and follow on site what is going on in both periphery and the central nervous system. So in parallel with that, if I may, in parallel with that, we have also an opportunity to monitor another patient group that we have had an opportunity to look at, and that's patients with herpes simplex encephalopathies. So herpes virus initiates in some patients a very severe degradation and inflammation in the central nervous system. And we now are monitoring a cohort, a fairly large cohort that's been collected over time, where we have multiple sampling points over the disease and when the patients recover. So we have now cerebrospinal fluid, the liquid that is surrounding the central nervous system, and we also have plasma blood samples collected over time in these patients. So what we're doing there, we are doing the same thing as we're doing on our post-COVID infected patients and our ME/CFS patients. We do proteomics, deep screening of all the protein biomarkers we can find in the body fluids. And in parallel, we do metabolomics, which is the small molecular patterns that are arising from the metabolism in the patients. And from there, we hope to learn what are the signaling mechanisms, what are the biochemistry behind the situation when patients are recovering from the disease, and also maybe learn about potential treatments.
I mean, I think one of the things that I like the most about this whole setup of research that you're doing there, Jonas, is the fact that you're looking at, ME/CFS, you're looking at the transition of COVID to ME, or what other people might be calling long COVID, and you're looking at this transition from HSE to these ME-like illnesses. And I think the benefit of that really is that you're going to be able to identify that whatever this ME/CFS signature kind of transitions to from these other diseases, there may be a commonality there, which may provide a good line of evidence to suggest that regardless of the trigger, regardless of COVID, regardless of HSE, these people end up looking like the same signatures as you see in ME/CFS. And that's something that I'm kind of looking forward to seeing how that kind of plays out.
No, I agree. And maybe I can give you a giveaway already that we are actually now, hopefully soon, publishing a follow-up paper also in Frontier Medicine, where we will actually allude to some of the treatments that has been used over years in intensive care unit patients, for instance. And we are trying to see what kind of mechanism that those therapies are using, and see if those will be relevant also for ME/CFS. And I think that's where we want to go in the end. We want to figure out what is going on, what is the biochemistry, what is the pathophysiology in the patients, but also we have to come to a treatment. And I think that is our goal, really.
Maybe I thought it'd be nice to kind of spend like a minute maybe updating where along. I mean, obviously you've described the projects that are ongoing, but whereabouts are you at this stage with the COVID-2ME projects and with the HSE, following the HSE projects as well?
So with the COVID studies, we are continuously collecting. So we are trying to collect as much material as possible now and get this temporary resolved sampling going so we can follow the same individual over time, which I think is crucial. But we are doing currently proteomics in the patients where we have cerebrospinal fluid, that's liquid around the brain. We do deep proteomics. We try to follow all the protein biomarkers that we can monitor with for looking for neuroinflammation, looking for cell damage, cell repair systems that are very relevant for this patient group. And in parallel, we do this metabolomics, the low molecular weight screening, both in cerebrospinal fluid, but also in blood samples that we are collected. And with the HSE patients, we are already done with all the proteomics. So we're already done, and we're actually doing the final data evaluation now, which is very intriguing and also very complex, as you may understand.
Yeah.
So, you know, thousands of proteins that are going to be measured and correlated to clinical situation. And we're also right now doing the metabolomics. So we will be able to compare also what the protein picture is and how the metabolism is related, both to the clinical situation, but also to each other, how proteins are regulating metabolism and metabolism potentially regulates proteins also. So it will be a very unique possibility here.
Yeah, I mean, it would be interesting as well to seeing the relationship between cerebral spinal fluid and the blood plasma metabolites and proteins as well, as you're looking at that. It sounds like obviously the HSE work may be coming out, you know, sometime in the very near future, and the COVID research is also underway with the assays occurring as we speak. So that's very exciting news. And it sounds like you've been pushing pretty well ahead there in Sweden over the past year. So, I mean, that's fantastic to hear.
And also, we have, of course, another track that we are following in parallel. We have everything has to go in parallel here to keep up the speed, basically. As some of you know, we published a paper last year also on the autoimmunity and the auto antibodies that we have monitored together with Carmen Scheibenbochen in Berlin in our Swedish cohorts in ME/CFS. So actually what we are doing in parallel now also is to see if we see the same kind of pattern in early stage of patients with COVID or with HSE, actually, to see if it is one part of the explanation could be that you cause onset of inflammation due to the initial viral infection, and as a side effect, a negative side effect, you get some sort of autoimmune reaction. And in some patients that may end up with an autoimmune antibody that may cause problems. So I think we have that we haven't dropped that idea yet. So I'm looking into that in great detail right now.
Certainly. Do you see any point in the future where that type of work can kind of come together with the work that you're doing with HSE and COVID or anything like that?
Yeah, definitely. So we try to learn from the different materials we are collecting, and I think we can, since we know it's a viral onset in HSE and COVID, and to a large extent we know that many of our ME/CFS patients present with an initial infection when they debut in their disease. So I think we can learn a lot from these parallel tracks.
Certainly, I think it'd be interesting to know, at what point along the line that autoantibodies start to show up, or autoimmunity starts to show up within these patients, whether it's quite early on or whether it's something that happens far along into the disease of ME/CFS. So I think there's plenty of possibilities happening over there in Sweden. Yeah, I mean, it's been fantastic to hear about all this type of research that's going on. And I think obviously you must be pretty excited with all these things that are coming out this year. Is there anything else that you're particularly excited or interested in explaining to the people at home?
I think we have there's a lot of things going on. So one thing that our PhD student is working intensively with is the method development. And of course, what we need to do in this field where we are sort of pushing the limits for technologies and measurements, we always need to improve the methodology. And that is ongoing work, and it will be continuous work, I would say, during the whole PhD program for our students. But I think it's very important to understand that there is limitations in all technologies, and we have to improve them to make them more accurate, more precise, giving the right information, because the methods, the measurement methods are sort of limiting what we can see, and it's also biasing what we can see, so to say. I think that's a very important task for us.
Yeah, I think it's probably like less exciting, but it's definitely very important, I think, in some of the research concepts, and I appreciate that a lot. I know that you've been working on certain panels in pathways that are probably of great interest to ME/CFS within that, and trying to develop methods for that. So, you know, it was interesting to listen to that. I know you were developing methods, I guess, to measure these hormones. And so that's something, as an example, that you've recently developed that method, and now you're able to look into that hormone work a lot more thoroughly. And that pertains, I guess, to the initial comments you made at the start of this talk in regards to your hypothesis and how the hormones relating to this inflammation system. So this is how everything kind of continues and gets started, yeah, you can see kind of science in motion. I think, in terms of like bringing it back to that hypothesis, because I thought there was some interesting aspects as it related to how the hormones are related to the neuroinflammation, and there was a nice little cycle that you had, a simplification cycle, I think, that involved, you know, the hormones relating to oxidative stress and inflammation markers. And a few of those oxidative stress things were nitrosative stress as well, which is probably something that a number of people have probably read in the literature in relationship to ME/CFS before. So I thought that was interesting that you were touching on a few of these common things that we've seen in this disease as well.
No, definitely. And I think there's, of course, a lot more to do there to understand it's very complex regulatory mechanisms. But one of my tracks is, of course, the neuroinflammatory part. And I think if we can understand why this low-grade neuroinflammation seems to occur and what is the reason for that, in combination with lack of energy and oxidative stress and nitrosative signaling, etc., etc. I think we are onto something there. I think difficulty to measure sometimes, but I think we have to do our best. I would like to add also, what is important also, and that we are also spending quite a lot of time on right now is when it comes to methodologies and measurements that we are combining and also comparing different measurement tools. So I think that is also very important, to not only to validate methods to each other, but also to complement each other. So we get certain possibilities with certain methodologies, and other methodologies will maybe very sharp on certain things, but lack the sensitivity for some of the targets that we have. So I think a combination is very important, and collaboration in that sense also extremely important.
I would definitely agree with that. And I mean, obviously some of the things that I think are really interesting to me is, I mean, obviously in this hypothesis you described it as a circle, and I think that's an important aspect to understand, that when we're looking at all these different pathways, that it really is kind of like a cycle, and it's not always trying to look for cause and effect. The cycle is, I guess, the thing that we're trying to look at in its entirety. And then looking at these kind of repeated measure studies, or these longitudinal studies in regards to or these cohort studies where you're looking at these diseases and how they may transition to different types of recovery. I think that's an area where we could potentially start looking into this cause and effect aspect as it pertains to how a cycle can kind of transition and change over time. So yeah, the culmination of everything that you're doing is really, really interesting. I think it's going to be it's an excellent way of looking into ME/CFS and understanding it better. And I have a lot of hope for the work that you're coming out with at the moment and into the future. So thank you very much for taking the time, I think, to speak to everybody about this. It was very much appreciated, and we'll catch up soon.
Thank you. All the best to all of you.
I'm pleased to introduce our next speaker, Dr. Wenzhong Xiao, who is co-director of the OMF-funded Harvard ME/CFS collaboration at the Harvard-affiliated hospitals. Serving on OMF's Scientific Advisory Board, Dr. Xiao is also the director of the Immunometabolic Computational Center at Mass General Hospital, Harvard Medical School, and leads a computational genomics group at Stanford Genome Technology Center. His research focuses on the interface of computation, genomics, and medicine, and in a moment he'll go into detail about the exciting research projects happening at the Harvard collaboration.
Hi, Wenzhong. Thank you very much for coming on to give an interview, give an update, sorry, I should say, for the ME/CFS community here through this through OMF research that you're doing. I thought we could cover maybe a number of topics. You're the big data analysis expert that we have, and I thought we'd start with kind of my first impression of you in 2017, I think, for the Stanford Symposium, when I saw you speaking about the severely ill patient study and the large amounts of data that you were handling. It became clear that you were dealing with massive amounts of data and very new technologies in the way of analyzing statistics as it pertains to big data. And so coming on from that, I guess I thought it was very interesting, the approach in terms of trying to generate as much information as you could for ME/CFS and to develop more understanding and information about the disease. I thought maybe we could start talking about maybe newer projects that you've been working on in terms of the data analysis component. One in particular I thought of great interest was some of this work with generating data from David Systrom's samples. And so maybe I thought you could maybe describe what they are and what that is, and what type of data you've generated, and where the analysis is up to on that.
Yeah, thank you. So I think the invasive CPAT study that you are referring to is one of those studies I think would potentially generate important insight to this mechanism in ME/CFS. The reason, as many of us know, is that ME/CFS is a complex disease involving a lot of organ systems. And using this invasive cardiopulmonary testing, we can simultaneously identify the dysfunctions in the physiology of the patients during exercise, as well as getting the molecular signatures from the circulating blood of the patients during and after the exercise test. In particular, we're looking at both the pulmonary blood as well as the arterial blood, which other technologies currently cannot easily have access to this type of information. So far, Dr. Systrom's lab has done hundreds of ME/CFS patients, as well as I think at least a couple thousand of other patients, for example, patients of heart failure or patients with pulmonary diseases. So that provides a range of diseases that we can learn from and compare and contrast with the ME/CFS patients. So far, we have done the cytokine analysis of the pulmonary and artery blood of the ME/CFS patients before, at the peak, and after exercise. And from there, the most striking finding that I think we found is that there's a significant increase in the arterial blood, those pro-inflammatory cytokines, comparing to the venous blood, which again, using other technology, it would be hard to get this information in those patients. And in particular, this increase is actually in response to the exercise itself. So we're still trying to figure out exactly which tissue, because when we think about the arterial blood versus the venous blood, the only organs in between are the lung and the arterial blood vessels. So we're trying to figure out exactly where these cytokines were generated, and that potentially can provide us new insight on how we might be able to potentially control this cytokine inflammatory response, and therefore controlling potentially post-exertion malaise. So that's one, I guess, exciting results that that we have gotten so far. And most recently, we have gotten the metabolomics data and the proteomics data from also the arterial as well as the venous blood of these patients. And our preliminary analysis shows hundreds of metabolites seem to differentiate between the patients and the controls at the peak and after exercise. And as we know, during exercise, the majority of the blood would go through skeletal muscle in response to exercise. So this might reflect something that's changing in the muscle of the patients comparing to the controls. So hopefully we can have more to report next time.
Yeah, that's really fascinating. I think obviously you spoke on the power of sampling within that that study with the invasive cardiopulmonary exercise testing, where they're effectively riding a bike, I think, and they have catheters collecting blood samples from the vein just before the heart, and then I guess the arteries just after the lung. So that's a kind of very unique type of sample that you're able to capture, and I guess looking at the difference before and after the lung and the heart, I think was kind of fascinating, that you were able to see a different signature in ME/CFS patients, maybe indicating there's something creating cytokines somewhere along there. Pretty very interesting. I guess it's also, for other analysis that you're doing at the moment in terms of research, probably similarly to what you're working on here in terms of looking for cytokines, metabolites, some protein markers as well, would be some of this COVID to ME study, this collaborative research study that's happening, looking into the clinical trajectories of COVID patients. And as they get to what makes people go on to develop myalgic encephalomyelitis instead of recovering as they normally would. And that's kind of, as I can tell at the moment, some of that data is starting to trickle through. Where are we at with that research project?
Yeah, that's a terrific question. So I think the study is going very well as a result of this multi-center collaboration between the Harvard Center and the center in Uppsala in particular. And we're receiving samples and data from Jonas Bergquist group on the COVID patients, as well as patients with related diseases. As you probably stated earlier, COVID provides a unique opportunity for us to actually understand the disease process for ME/CFS, because as we all know, a substantial percentage of post-COVID patients, unfortunately, would have a high chance of getting into ME/CFS. But on the other hand, in this case, we know that the etiology and the timing of the infection, which trigger this disease process. So that sort of provide us a unique opportunity to study the development of ME/CFS. In this particular study, we would also have access to the CSF sample, the cerebral spinal fluid sample, which is a unique tissue type for us to study. So right now, we looked at the proteomics of patients versus controls, and you can clearly see this neural information in those patients in the acute phase. The question obviously is, when the patient eventually, if the patient eventually develop ME/CFS, whether at that point we would still see a clear neural inflammation in those patients. That remains to be seen, but at least in the first 2 months, we see a clear difference in the proteins in the CSF between patients and controls, which is a exciting finding, and hopefully this can help us to identify the disease mechanism and eventually treatments in these patients.
Yeah, I mean that's kind of that's the holy grail. That's what we're really after at the end of the day, and also just looking for predictive markers and some maybe understanding that etiology. I think would be fantastic.
Absolutely.
To go in. So I think also, I mean, I guess on that note of treatments, I think one of the more interesting sort of work that I've seen you actually been working on has been this look at the network analysis for treatments. And I guess this is kind of, I'm trying to identify, it's a complicated concept, but effectively what you're doing through data analysis is trying to identify treatments for ME/CFS, potentially repurposing of those. Could you maybe go over a kind of a description of that and some of the interesting avenues maybe that could potentially go down from your findings?
Sure. So by the way, that's obviously also a collaboration with you and your center. I guess the overall idea is to essentially compare the data that we have in ME/CFS with the data that people have already, or scientists have already collected in other diseases that are better studied than ME/CFS, for example, heart failure or neurological diseases. And as we all know, because ME/CFS is such an understudied disease, the data that we have is largely, scattered. So there's a need to actually integrate data from multiple studies each individual, one of those studies seem to be of a small sample size and might only look at one aspect of the disease because obviously funding issues. And then compare this pattern with the patterns that are in other disease studies people have already established, and see which part of the signature in ME/CFS is similar to the part that people already know in other diseases, to see essentially whether there is a subset of well-known diseases that look similar to ME/CFS, and if so, whether there are proven drugs that are effective in those diseases that we might be able to consider in ME/CFS. So that's our current focus. And a step further would be to look at actually the combinations of symptoms in subsets of ME/CFS patients. For example, if the patient is mostly affected by, let's say, sensory disturbance, would there be, other diseases that would also show similar combination of symptoms, and whether some of the drugs that are used in those diseases can be, as you mentioned, repurposed or tested in this subset of patients. So that's another area that we're currently looking at.
Yeah, I think these are very, very smart way. I mean, it's, as you describe it, to be quite, in a simple way like that. It's very, very different when you see it on a page and the complexity of the information that you're kind of creating here, and the pathways. I'm sure it'd be great, obviously, to see that in a presentation sometime soon, in PowerPoints and all that sort of stuff. But I think people get the picture in terms of how it's, the value of what you're doing. I think getting that across is really fantastic. I think that work is very important. In terms of, I guess, other research that you've been kind of conducting with ME/CFS, beyond the research, one of the key aspects is kind of this maintenance and the creation, I guess, creating the creation and the maintenance of the data center that we all utilize to kind of put data up and share with each other, but also able to kind of get insight from other people outside of us in the broader community, and also get other people within the science community to kind of upload their data if when possible. I think that's a really important resource for us working together on this disease. And then also, obviously, you've started developing the REDCap component to that, which is to try and integrate some clinical data. So I just thought maybe you could speak a little bit about that as well.
Sure. So on the data center side, as we just discussed, there's, I think it's essential for ME/CFS research, just because the current studies are limited in sample size, as well as the dimension of analysis, because again of this lack of funding. So each study can only typically look at a few patients and look at only one aspect of the biology. So there's this essential need to actually combine data from multiple studies in order to get a better picture of the disease. And this data center is established to facilitate this kind of collaboration. So right now, as you know, data from multiple OMF-funded data centers, research centers, and OMF-funded projects would put the data into the data center before the study is published. So that enable researchers in the entire community to have access to the research data and freely exchange their thoughts on the data and the findings before the publications. And I think that's, you know, I think that's essential for research in ME/CFS, getting new insight in this disease. And the second aspect of that is, since we're trying to combine what we know in other diseases together with what we know in ME/CFS, getting this data as well as the interactions with other diseases also in the data center would facilitate us to generate hypotheses and test those hypotheses quickly, and as you just mentioned, considering the candidates for clinical trials. And the Red Cap project is established to support basically this clinical study and try to enable the recruitment of patients using a uniform standard of the inclusion and exclusion criteria. And as we know that getting consistent patient recruitment, especially for complex disease as ME/CFS, is critical for the results and data to be compared across different studies. So that's one of our focus for this Red Cap project. The other focus of this is to enable generating surveys across multiple research centers, and so we can actually survey patients and get information and pull information together.
Yeah, I think that's a fantastic resource and very, very instrumental to the work that happens around all the research centers, I think, in OMF to some capacity. Wayne, I think you're doing fantastic work, and thank you very much for updating the community on that work that you've been doing. Cheers.
Thank you.
No worries.
Thanks.
Our next speaker is Dr. Alain Moreau. He's one of the newest members of the OMF Scientific Advisory Board and is a full professor in the Faculty of Dentistry, cross-appointed into the Biochemistry and Molecular Medicine Department in the Faculty of Medicine at the University of Montreal. Dr. Moreau is also the director of the OMF-funded Montreal Collaborative Research Center in Canada and will now be presenting the exciting and innovative research projects that are underway.
Well, hello, thank you very much, Alain, for coming on to give the good people in the community OMF update of the kind of the research that you are doing at the moment for MECFS. Thanks for being here, and I remember the first time that I actually saw you talk about the research, and that was in this Montreal symposium that you, I think, had a big part in organizing back in 2018. And I think one of the most interesting things about what I at least stuck out in my mind was the fact that you're utilizing this technology to create a mild stress response in the MECFS patients that you were measuring to look, or really to characterize post-exertional malaise. And I thought what was really clever about that is that you've created this standardized way of creating the stressor and using that as a comparison rather than getting each individual patient to stress of themselves in a way that might be differential. And I think in that capacity, what you've created there may be a useful way to for many other researchers to kind of look at post-exertional malaise, if they could use it in a consistent way, and even to kind of work maybe even diagnostically or in any way of monitoring patients or monitoring post-exertional malaise in patients for all different types of studies or clinical trials. So I thought that was super fascinating. And I thought maybe you could start there and kind of describe that device and some of the research you did around that time.
Hi, Chris. First of all, thank you for the opportunity to be interviewed by you. So yes, we develop a stress test to reproduce or induce in a very standardized way post-exertional malaise. First, when I embarked myself in my research team in this new field for me, I listened to patients, and patients really insisted to the most important aspect of the disease, the post-exertional malaise. So whatever I intend to develop at that time must be centered around the post-exertional malaise. So that's why we reuse a technology that we tested in the past in the lab by using this mechanical stimulation using an inflatable cuff that you apply on any body part, but in our case we use the arm because it's more useful. We developed this stress test, which seems to mimic a kind of exercise response to the patients that leads to the induction of post-exertional malaise. So first, after over 200 participants that we tested so far, we clearly demonstrated that yes, we induce post-exertional malaise. Yes, the patients are a bit more sicker for a few days, but we are doing in a way that relatively safe. So we didn't have to call an ambulance or to hospitalize any individual following this test. So that was one of the priority number ones, to be able to do it in a safe way. But as you know, if you induce post-exertional malaise in an MECFS patient, they will be a bit sicker. So we reach, I think, safely that point. Secondly, by the application of this stress test, we hope in a single test to reveal what exactly is happening in a patient. So, and as you know, the patients are coming from different type. So clinically speaking, they are very heterogeneous. The trigger factors that led them to MECFS is different in some cases. We know that the vast majority develop MECFS following by a viral infection, but even though the virus can be different, and there are other causes that led to ME/CFS. So that's why we need to figure out something that can be able to, independently of the trigger, independently of the illness duration, independently of the medication taken by each individual, we need to perform a pragmatic test that can be able to decipher the response and lead us to the real molecular signature that is associated with ME/CFS. So we developed this test, and each individual become his own control, because we have, after 90 minutes stimulation, we draw blood, and before a stimulation, so we have blood, so we can compare changes between each individual. So which is one important point. The second important point for us at that time was to be able to reach out the most severely affected individual, which are the individuals that are rarely being tested because they cannot access to the clinic. They are incapacitated. So 85 to 90% of our participants are housebound. So which means that we are sending the clinical nurses at home to do the testing. And that's why all the apparatus we are using are portable, and they are relatively cheap. So it's not very expensive a machine. So which allowed us to do the test. And of course, we are measuring different factors. So we start, as you know, by measuring circulating microRNA. There are very small molecules that are non-coding, but they are very powerful regulator of many genes. So normally you might expect that one microRNA could regulate up to 200 different genes, and some gene can be regulated by more than one single microRNA. So very powerful regulator. They are very well studied in molecular medicine as a biomarker for different diseases, especially for complex diseases, and I think ME/CFS is a complex disease. So to make a story short, and following our publication in scientific reports, I think we were able to stratify patients in 4 subgroups that are associated with some symptom and symptom severities. So right now the test is accurate up to 94% to predict blindly who is suffering of ME/CFS versus being a healthy control. And what we can do also is the test can also predict the therapeutic response. And I think this is the future of the test, not only to be able to identify symptomatic individual with a precise and rapid diagnosis, but also to try to establish a kind of prognostic. Should I put this individual, for instance, on Amplygene, or should I select something else? So this, I think, can make a kind of revolutions that are wait to be replicated by others, but this is where we are going in the next phase of this development.
Yeah, I mean, there's a lot of information there. It's a fantastic study, and I do recall, that publication you had in Scientific Reports, which is a fantastic journal. I think that the interesting part about some of this work was the microRNAs, and I'm not sure whether everyone is familiar about exactly what they are. I think you went into a little bit of a brief description, but I know from myself, and I kind of think I follow a similar logic in terms of the research with yourself, in terms of looking at symptoms and metabolites, which metabolites are kind of an expression of genes, but also an interaction with the environment. And I believe that microRNAs probably fit a very similar bill to that. It's just a different area, but they're both very closely associated to the phenotype of a person, which makes sense when you're characterizing symptoms. So I thought maybe, we could touch a little bit more on this microRNA work. I think that if there's any detail, I guess maybe describe how the microRNAs are generated, and potentially what they could be, also what the impact of them is on the host.
Yeah, so as you may know, 98% of our genome was so-called junk, a few years ago, maybe a decade ago. And right now we know that only 2% of the genome code for something, okay, for gene that will produce a protein or a peptide. And we and others realized that this so-called junk code also for many non-coding RNA. So we have two classes, the long one, which are about 200 nucleotides in length, and you have also the smaller one called microRNA, which is about 22 to 26 nucleotides. And the way it works, and I don't want to go in the detail of the biogenesis of both because it's way too complex, but the points to understand is, especially the small microRNA, they are attaching to the messenger RNA, which will be eventually translated in the what we call the ribosomal machinery that will read the copy from the DNA that we call it messenger RNA that will lead to a protein. And by attaching at the end of the messenger RNA, that blocks the decoding. So the machine, the photocopier is blocked. Okay, so in a non-technical term. And also simply the fact to attach to the messenger RNA, the messenger RNA don't like to this type of duplex, and often that brings some signals to degradation. So sometimes you see the messenger RNA start to be degraded, there will be no protein, or we jam the photocopier, if you wish, and the ribosome cannot decode the protein. So such attachment, and you can have many sides, so that can really block completely the translation, and you will see a dramatic decrease in the protein that should produce. So if it's an important protein, you will have a dramatic effect that can lead to more fatigue, to brain fog, to orthostatic intolerance. So altogether, they may be the cause of the, let's say, hallmark symptoms of ME/CFS.
Yeah, I mean this is kind of the interesting research that I guess one of the more interesting aspects of what you found was actually those subgroups, and I think that's kind of important because you're thinking about the disease in different ways. It could be produced or in different capacities or in different kind of packets, I guess, for the different individuals involved with ME/CFS. And so I think if you can kind of subgroup them, that might be a better way to kind of understand their biology. I think because the diagnosis is based on symptoms, there's no guarantee that the underlying biology is going to be exactly the same. And so we have to kind of think
I
in a
agree.
way
I agree that with the way. Yeah, sorry. I agree with the way that you summarize this. So we and the clinicians in particular already noticed that we are facing to a very clinically heterogeneous disease. So technically, ME/CFS, even if we regroup all those symptoms, is not truly a single disease or single entity. So there's a kind of spectrum of the symptom, and that makes a lot of sense because there are different triggers that being involved that leads to the phenotype or the disease. So now having more objective molecular markers that can sustain this view and having this group, that will allow us to have a more profound understanding of the pathophysiology of the disease, but also to better select maybe some therapeutic options, so and prevent to use something that it's quite unlikely that the patient will respond because having an elevation of this or a group of macroRNA, the target is disappearing. So how I can connect a drug on a target if the target is not there? As the opposite, maybe some microRNA by decreasing the target pop up, and now you can now use a drugs on this very specific group. So I think if we understand the capacity, having a good or better knowledge of those markers, open up more therapeutic options using existing drugs. So I'm not even thinking about developing new drugs, but using existing drugs. So that can have a tremendous impact in the way that researchers and clinicians will, in the near future, design clinical trial. It's like now in oncology, in oncology, you cannot launch a drug anymore if you don't have a theranostic. So a test that will predict who will respond to your drug and who wouldn't. So the FDA and other regulatory agency won't authorize your clinical trial or the development of your drug if you cannot select the patient. So I think what we are doing and other colleagues are doing is to bring ME/CFS in the era of precision medicine and to apply this, and that I think that will increase the success of the next clinical trials, and I think it's very important.
And so I guess one of those subsets or one of those subgroups that you found was quite, I mean, a number of those have been quite interesting. Did you want to unpack anyone in particular to kind of go into more detail about so maybe some of these treatments or some of the aspects of that may be
Yeah, just to sum up about, I will give you an example of what we refer to cluster 3. So cluster 3 regroup mostly ME/CFS patients suffering of orthostatic intolerance, often leading to tachycardia to compensate the hypotensions. And by now having this stratification of patients, we look again about different biomarkers that we measured before, like different biochemical marker like thrombospondin 1. And thrombospondin is of interest for us because it's a powerful vasoconstrictor. So in cluster 3 at baseline, you have a high level of thrombospondin, which means that your arteries are constricted, which reduce the brain blood flow. And after the post-exertional malaise induced by our stress test, there is a drop in the thrombospondin level, often around two-folds of the initial 90 minutes before. And that is sudden to open up the artery because now you create a vasodilatation, and this surge of the blood flow will create a hypotension. And this is when often the heart will react. So the pump will activate to compensate, and you won't like it because that creates a tachycardia. So knowing that, we can now pinpoint this group of patients in cluster 3, as opposed, for instance, patients in cluster 4, who they are going in the opposite direction. They start with much lower quantity in their blood of thrombospondin, but after the post-exertional malaise, the thrombospondin increase, and now vasoconstrict, and now this is where brain fog is creating. I reduce your brain blood flow. So very easy to explain, very easy to demonstrate by measuring those physiological change using the smart biometric vest like Exoskin. And also, we saw the change in the brain oximetry with using another technology that confirmed that we saw the decline in some patients on the oxygen level that suggests that we reduce the brain blood flow as part of the stress test that is inducing post-exertional malaise. Now, what I'm doing with this information is that there are ways to regulate the level of thrombospondin. There are ways to block the receptor where thrombospondin is acting, and there are non-pharmacological and pharmacological treatment for that. So that's why I think that will be the next logical phase for us to launch specific clinical trial on selected individual using our tests to select them, and now to prove whether or not this is efficacious or not. So we need to do a randomized clinical trial to do it, but the big difference is we will select not the whole bunch of ME/CFS patients because you will fail. This is a very select and specific subgroup. So I think we will increase the likelihood of success doing that. And I think this is why I think it's very important.
Yeah, I mean, I agree 100% that. I mean, that strategy that you have taken, I think, is extremely clever as a way of kind of getting around some of the complications we've faced. I mean, there has been a few clinical trials on ME/CFS for different drugs, or one very famous one. And I guess you can tell that there are, benefits for some and not for others. And then this is a way of kind of adding some science to that, having that prediction, and then going into the trial, you can involve that prediction model that you have, or using your tests to identify those patients who will then very well more likely respond to this drug. I think that's a fantastic way of doing it, because then you're going to get people involved the FDA can come through and approve and all that sort of stuff. So if you can go through, I mean, that process over and over, that would be great. And so it sounds like you're starting with the clusters in regards to thrombospondin 1 and relating that to certain type of treatments. I know another project that you've been working on is in regards to DNA methylation and its involvement in ME/CFS. Did you want to potentially introduce that project and maybe describe some small aspects of that?
Yeah, this is a sub-project in our research program called Maestro and Me, where we are looking at all the multi-omic aspect of the disease. And beside the microRNA, there are also other epigenetics effector called the DNA methylome or DNA methylation changes. So, and we decide to get more insight about those changes by studying several couple of identical twins, but discordant for ME/CFS, which means that one twin is affected and the co-twin is not affected. This is a very, very powerful strategy to set aside what is the genetics component of the disease and what is the epigenetics, so which is coming from the environment. So as you know, the genome of identical twin is they share about 98% of their genome. So we could easily exclude that if the disease is in one twin, this not is due to genetics, but mainly to epigenetic changes. So this is one thing. So, and thanks to Open Medicine Foundation and other association of patients, we were able to collect over 15 couple of discordant twins around the world. So, and because they are localized everywhere in the world, we cannot draw blood easily and send nurses or try to locally organize a clinical setting to receive them. So we use the collection of the saliva. So the saliva is a minimally invasive collection. Everyone is able to spit in a tube, seal the tube, and send the envelope back to Montreal. And very, very stable when the saliva is in the tubes, even at room temperature, so which is one of the advantage. And the second thing that beside the pragmatist, again, the using of saliva, the methylome from the epithelial cell present in the saliva is mimicking more closely what is happening in the brain and the muscle than what you can see using blood cells. So I'm not saying blood cells are not important. In fact, blood cells are very important. We know immunity is involved, but you have a stronger heterogeneity. You have different level of different blood cells. So when you combine all of them, that might change quickly. And others before me, I'm thinking about Patrick McGowan in Toronto, who studied very well DNA methylation using PBMCs, or others in Australia, your colleagues that study NK cells methylation more specifically. So because methylation patterns are specific for a cell type or a tissue, so we have to be aware about that. So I think we are the first using saliva in the context of ME/CFS, of course, and because it mimics or seems to reflect what is happening in the brain and muscles, so two important tissues for ME. So we did again a gamble to continue to use that, and we used the twins as a discovery cohort, and now we are in the process to validate a candidate gene because we saw very specific changes. And again, the idea here is not only to see the changes to establish some profiles, to predict disease severity, but also to target the affected genes. And I will give you just one example that I think is very exciting, is about, we saw in the twins, in three couple over 4 of the twins, so all affected twins presented a DNA permethylation in a gene encoding for the key glutamate transporter. So, as you know, others before me presented some evidence that you have increased glutamate in the brain and sometimes in circulation, also in the muscle part. So it's not present everywhere in every patient, but in subgroup of patients. Now we think we have the possibility to identify the individual where glutamate is more prevalently accumulating in the brain and muscle due to the measurement in the saliva cells. And also, the interesting part is there's a way to, let's say, because due to the DNA hypermethylation, that gene will be less expressed. So we will have less glutamate transporter and it cannot be removed. So which explain why it's accumulating and creating fatigue, brain fog, and other effect. So now we have drugs that are being tested for different disease that are safe and wake up that gene and can bypass the DNA hypermethylation. So again, the exercise we will publish soon is to showcase what is the prevalence of this gene. Is it really downregulated due to DNA hypermethylation? And at least we hope to make the demonstration in vitro that we can use different type of drugs to wake up the gene, if you wish, and to reverse and increase the capacity to prevent glutamate accumulation. So again, our goal is not just to look for biomarker, but to look for actionable therapeutic target that will improve the health of specific ME/CFS patient.
Yeah, I mean, that's a fantastic approach, and I really, really appreciate that. I mean, all the type of research that you're doing, I think, is very cleverly targeted at trying to get treatments for patients with ME/CFS, thinking of ways in which you can kind of categorize patients so that they can get more effective treatments. I mean, this is very interesting work, Alain, and I wish you all the best of luck with that. And thank you very much for the update that you've given to the community today. And we'll
Thank
look
you,
forward to
Chris,
hearing
very
more.
much for your interview and the opportunity to interact with the you and the patients that will look at this interview. Thank you.
Fantastic. Thank you.
Next up, I'm glad to introduce Dr. Christopher Armstrong. Dr. Armstrong is the director of the newest OMF Collaboration Center in Melbourne, which was established in partnership with Emerge Australia in 2020. Chris first started his ME/CFS research at the University of Melbourne through his PhD project that applied metabolomics to study ME/CFS. Now back in Melbourne, after spending some years as a visiting scholar at Stanford, Chris is guiding OMF's exciting new collaboration in Australia. His initial research is focused on an individualized approach to understanding ME/CFS. Please join me in welcoming Dr. Armstrong from the Melbourne collaboration. Chris, thanks for giving us an update. How has the transition from the US to Australia been?
Thank you very much for having me and allowing me to present an update like this. And thank you for OMF for holding these interviews of the CRCs. I think it's important, obviously, to communicate to the community as much as possible. And I'm hoping this is the first of many to come. So just starting with the transition back from the US to Australia, I think that was a lengthy process at the end of last year, but we managed to get back into Australia in December last year and started up with the University of Melbourne in January of this year. Obviously, I'm the director now of the Melbourne ME/CFS collaboration. And in that role, we've really started to try and get as much collaboration happening within Victoria at the moment as possible, and obviously expanding through collaborations worldwide as well. At the moment, we've gone to first point of call has been to actually bring on new staff. So it's not just myself. So we have a PhD student and a postdoc who have joined the group here at the University of Melbourne. We've been trying to set up a lot of these projects that we have. We're starting from scratch, I guess, for a number of these. That has included a lot of putting together ethics applications, getting approval for these studies, but also going through the protocols and setting them all up and optimizing, I guess, those protocols for the projects that we'll be doing. We have started a little bit of a pilot project in collaboration with Monash in regards to looking at Monash University in regards to looking at a diagnostic but more of that information will come, not in this update, but the next one as we get more results. We also have a couple of longitudinal studies that we're starting up. So one with adults, but also with pediatrics at the Royal Children's Hospital, which is just down the road from my institute, the University of Melbourne. And so at the moment, we're very optimistic for a big year ahead. We have no restrictions here in Australia. Everything's back to 100% normal as far as I can tell. From that, we can probably expect to have a full year of research unobstructed, I guess. And so that's kind of the big benefit for being here at the moment. But also, I think that hopefully we can get more results out as soon as possible. I think that would be obviously music to a lot of patients' ears.
You mentioned that you're setting up a number of longitudinal studies. Can you explain what they are and why you are taking this approach?
Yes, we do have a few longitudinal studies, I should say. So longitudinal studies are effectively repeated measure studies. So instead of having a case versus control population, which is what we might do more commonly in ME/CFS, and a lot of studies have worked on that in the past, looking at a cohort of ME/CFS patients and a control cohort, and looking at the differences between them. So the difference for a longitudinal is that we're trying to look for more of the repeated or some of the dynamics of the disease within the individual. And so you could potentially do that, still looking at case control, looking at some of the changes within an ME/CFS population versus a control population or another disease population. That's also a way of doing a comparison. But for what we're doing a significant number of repeated measures in the individual, and we're hoping to use the individual as their own control. And what we mean by that is getting a number of samples along a period of time to get a kind of a baseline, and then also get samples at the most severe points of their disease to have a comparison. So we're trying to compare severe exacerbated periods where they have extreme symptoms or worsened symptoms versus periods when they have less worse symptoms or relatively good days, as we call them. And so I guess that's a way of kind of understanding what might be going on within the individual to see if we can find biological changes that kind of represent what's happening when they get worse and what's happening when they get better. So the value of that, we think, is because obviously some of the major ME/CFS symptoms like post-exertional malaise are kind of dynamic symptoms. They're not the sort of thing that you can just say there's one point where you have post-exertional malaise. It's the exertion event getting significantly worse, and then a protracted recovery, I guess, from that. And so I think this is kind of the value of doing this type of research for ME/CFS, but also because this disease is kind of unknown and it may be heterogeneous. Yeah, so we consider it to be potentially a heterogeneous disease where you may have multiple different disease pathways underlying different patients with ME/CFS. That's a possibility. We still think, obviously, looking for a central component to the disease, but because the disease itself is outlined by symptoms and not with some defined pathology, I would say, it's still kind of not 100% known whether that all the people who are kind of diagnosed through the symptoms with ME/CFS do have the exact same underlying biology. And so that's why we kind of think of looking at longitudinal studies for ME/CFS patients, because we can kind of start defining individuals and then start grouping them, I guess. If ones who look very similar to each other, we can kind of group together and then more deeply characterize that group and compare to other ones as well to see if we can find any subsets of the disease.
It sounds like you consider this to be a heterogeneous disease. Do you not think there could be one cause for the disease?
Yeah, I guess it's not that we consider it to be a heterogeneous disease. It's actually that we don't know whether there is a single cause or there might be multiple. I guess by doing this approach, we're considering that there may be either way. And so we can kind of look for whether it is heterogeneous or it is one component to it. Personally, I think we've taken the approach that we do think there is a central component to this disease, but multiple pathways that lead to that central component. Because the component is so central, I think that means that there is probably more likelihood for multiple pathways to it. And so that's kind of the approach that we take. So I guess we would say we think it's mostly some component, obviously, underlying the common symptoms is probably consistent between patients, but then there's also going to be some variation. So I think that's probably the best way that we can kind of approach it at the moment, since there is no knowns at this point in time. However, of course, we keep an open mind, and as I say, that's the reason why we're doing what we're doing.
Can you describe the nitrogen hypothesis in simple terms?
Yes, the nitrogen hypothesis. So that's kind of what we do think is the central energy metabolism disturbance, I guess, that is common across ME/CFS patients. That's at least our hypothesis. We don't know that, but at this point in time, that's kind of the theory that we're working from. I guess to describe it in simple terms, maybe I'll start with a scientific explanation and then go for an analogy that might help to explain it. So trying to keep it as simple as possible. For energy production in the body, usually required to, I guess, burn carbon to create carbon dioxide. So the carbons will then effectively jump onto the oxygen that you breathe in. You breathe out carbon dioxide. That carbon being split off from metabolites creates these protons, or loads these protons in the mitochondria, and then you're able to utilize those protons in the mitochondria to pump through and create ATP, which is the energy molecule that you use for all types of reactions within your body. And so carbon is central, I guess, for metabolism, for energy, for oxidative metabolism, especially, or for all metabolism, actually, for energy production. But so what makes carbon interesting is that, carbohydrates and fats and amino acids all have carbon in them. And what makes them different, I guess, is that for amino acids, what makes them more unique is that they have the nitrogen molecule, whereas carbohydrates and fats do not. And so they're also a very useful form of fuel, I guess, for producing ATP. But in that process, the body has to remove that nitrogen from those metabolites, from amino acids. And so when you're more heavily using amino acids for ATP production, you're producing more of these nitrogen byproducts. And some of them can be quite toxic in certain types of accumulation. And your body has, or your cells have ways to remove that nitrogen from the mitochondria. But if you're more rapidly using it, you may not be able to, I guess, remove it as fast as you would like. And that's really the basis for the theory of this, which is that more amino acids are being utilized for energy production, or effectively more nitrogen is accumulating, or nitrogen byproducts are accumulating in ME/CFS patient cells when it comes to energy production systems. And so they are having a kind of a toxic effect. And so I guess you would say that the basic understanding is that through this mechanism, the production or creation of energy is actually creating a problem in ME/CFS patients. One of the most initial problems of nitrogen is the accumulation of what's known as ammonia, which can actually pretty much stall energy production pretty rapidly. And so the more that you produce energy, the more you're creating this ammonia if you're using amino acids, and the more that this ammonia accumulates, the more likely it is to pretty much slow down that energy metabolism, and pretty rapidly as well. And so we think this could be a kind of a basis for a number of symptoms with ME/CFS. Obviously, ammonia and nitrogen byproducts are also being a long literature history of being attributed to issues of cognitive dysfunction, brain, things that would indicate brain fog, but also you have encephalopathies and all that sort of stuff. This is kind of different, and this is not the sort of thing you would expect to find as high ammonia in the blood for our hypothesis. This is what we're actually indicating, is that you have, I guess, an accumulation of nitrogen within the cells themselves, okay? And so I guess if you were to use an analogy for that, if you have the sewage being indicative of the blood, and then you have a problem, I guess, at the sewage treatment plant, this is be more like what we think of like when you have an issue with your liver. Then you start to get, I guess, an accumulation of toxins throughout the sewers because the treatment plant isn't removing or those toxins. And so that would be like ammonia building up in blood. And that's when you have something wrong with your liver. You get elevation of ammonia. They can test that at the doctors, and usually that's to do with something like cirrhosis or some type of liver problem. I guess in this example, if you're having issues at the point of the houses, like in the bathrooms, or there's all the toilets suddenly start breaking, and everyone, like the sewage component of that being built up in the houses, you're not going to see it within the sewage plant. You're going to see it at the homes, and that's kind of like the cells, I guess, in this analogy that I'm talking about here. Some interesting aspects of this is more common in females. Females are more rapidly utilize amino acids than fats. That has a lot to do with actually female composition being higher in fats and lower in protein compared to males who are higher in protein and lower in fats. And a lot of that is to do with sex hormone differences. And so females are more rapidly utilizing amino acids already. And there are a number of things that can contribute to this, not just the increased use of amino acids, but also the decreased ability to remove nitrogen byproducts from the mitochondria or anything like that. And so that's kind of like the idea behind this hypothesis, I guess, more scientifically. And you may wonder like how we think that kind of comes about. We think it could very well be that there are genetic issues within nitrogen removal, or at least increased amino acid consumption. Pathways that lead to that may be certain dysfunctions in the use of sugars or fats, but also could be dysfunctions in the ability to remove these nitrogen byproducts as well that get produced. And so obviously issues in fats and sugars would lead you to use more amino acids. And so we think that is kind of a way of pushing towards the use of amino acids as well for energy production. And I guess a kind of an analogy I like to use, and I think it works pretty well for describing what we're talking about. You could have had a house, I guess, and you have a petrol generator as a backup for energy production in your home. Now you have on-grid power, and I guess you're going through your entire life. You have on-grid power, and there might be periods of time where you get shutouts of that energy, and you're going for the backup generator. Now I should add that under stress, you increase your production, or you increase the usage of fats and amino acids. So usually the body likes to use carbohydrates for energy production, but you increase the amount of fats and amino acids for energy production under a stressful condition. And so we like to think of this as kind of like a backup generator during stressful periods. You start to use your petrol generator effectively, different types of fuels to create energy, I guess, within your household. And then maybe it's working fine for little short periods of times. You have a cold, you have other stressful events, and the backup generator can do the job. But then suddenly, you have a blackout for a long period of time, maybe a month. You start using the petrol generator, and there's something wrong with it. There's always been something wrong with it, but you never knew because you never used it long enough to realize that there was an issue. And so what I'm talking about is whether you have genetic anomalies within that backup generator pathway, within the amino acid or fat metabolism pathway, and within the nitrogen removal pathway. So you're using this this petrol generator for a period of time. Perhaps it's spreading fumes into the household from the petrol generator, and you're starting to get quite unwell because of that. I guess that's the analogy, that we're running with in terms of a potential explanation for how this could crop up for people at a later stage in their life, not just something that they're born with. And so that's kind of, the best or simplest description of this. We have written up a lot more detail on this hypothesis and into a paper, which we will be trying to publish in the very near future. I'm sure people will be very interested to hear more about that.
That's an interesting hypothesis. How do you plan to test it?
Our plan for testing this hypothesis is actually going to come from trying to measure these nitrogen byproducts. So both in cells, so taking cells from MECFS patients and tracking the cellular metabolism, and specifically labeled nitrogen substrates. So we can feed the cells substrates that are specifically labeled for the nitrogen and look where the nitrogen goes through MECFS cells versus healthy cells. That's one thing that we'll be doing to test it. Also, through these longitudinal studies, we'll be checking through the urine. So most of the time you remove nitrogen byproducts through your urine. And so if we can track over time, it might be too subtle to track one sample, but we plan on tracking 24-hour urine, maybe over several days, and just determining if we can find a link between increased nitrogen output and energy usage that is more exacerbated in ME/CFS patients than controls. And so those are the two ways. One is, I guess, an in vitro measure and not incorporating environment. So in vitro is the cellular, growing the cells and looking for cellular metabolism. That won't tell us much about whether there are environmental factors. More, it's just going to tell us if there are genetic factors leading to potential increase of this nitrogen byproducts in the cells. The in vivo measures will be the longitudinal, where we're able to get a total output from the individuals to see if they're getting an increased output of these nitrogen byproducts, potentially from urine over time. So yeah, that'll be the main ways that we're going to be testing that.
We'll all be interested to see how that turns out. Thank you for taking the time to update the community, and we look forward to hearing from you soon.
No problem. Thank you very much for having me. We'll check in soon.
In closing, I'd like to sincerely thank our wonderful directors for their informative presentations, and to Dr. Maureen Hansen for her excellent job as a moderator, and to each and every one of you who joined us to hear more about some of the projects that your donations are funding. I'd also like to send a special thank you to all the patients who volunteered to participate in these research projects. We could not do this without you. Our understanding of ME/CFS and related diseases is advanced only by the people who volunteer to accelerate our research and to be a part of the solution. Thank you very much. It is through your support and generosity and interest that we're able to accomplish this important work. And for that, I am truly personally grateful. We have a long way to go, but we work every day to uncover opportunities to attract resources and level the research playing field. Today, we're kicking off our annual May Momentum campaign. In recognition of May ME/CFS Awareness Month, this campaign is a special effort of ours to join others in increasing awareness of ME/CFS and to raise funds to continue accelerating this research. I sincerely hope that you'll join our worldwide community to support this urgent research, growing now sadly with many long COVID patients transitioning to ME/CFS as well. You can donate online, create your own fundraiser, help to spread the word and spread hope. Your support in any capacity makes a real difference, and it helps us sustain the momentum year-round. I hope that our open house has illuminated the groundbreaking research being done by our remarkable directors, working night and day to find an end to ME/CFS. Even in light of the challenges we all face through this pandemic, we have continuously found ways to keep collaborating and accelerating research and moving forward. I think this is a true testament to the dedication and the commitment of each and every director that you heard from, and the wonderful teams of supporting researchers working with them. It is my wish that you leave this event with a feeling of hope for the future of ME/CFS and related diseases. Rest assured, we will not give up until treatments or a cure are found. Thank you very much.